able to see the slides?
Yes, sir.
Yes, sir.
Yes. Yes. So good morning to all of you. So I'm not going to discuss much about the amino acid because you people already know about the amino acid in your first year, second year, or third year. So I'm going to discuss few things about the amino acid. Reason is that the subject is protein engineering course, so that you should know little bit about the you should not little bit should know about the characteristics of amino acid. So just one basic today I am going to discuss. So you people, uh, first year or second year, you have studied amino acid, student?
Yes, sir.
Yes, sir.
Yes. So okay. So just I am giving a small overview about the amino acid because it is mentioned in your course also. So the in your syllabus. So we know that base if you want to amino acid better you can remember CORN. CORN is a acronym for COH, the main chain carboxylic acid group, the R group representation of side chain, NS2 nitrogen of the main chain amino group. So basically this carboxylic acid, NH3 and hydrogen is common, but R is, if R is changed, the amino acid properties has been changed. If people know 20 amino acid, some are essential, non-essential amino acid. I am not going to discuss all these things here. So there are 20 amino acid. One thing that I am going to discuss here, that you have to always remember two things that maybe it will come sometime in during the course. The carboxylic group pi is equal to carboxylic group pk value is 2.2, sometime 2.3, some book 2.4, amino acid is 9.4 or 9.2. These things we have to remember few things. All physiological psi amino acid are charged and shown to left. They can act as either acid or base. I'm not going to discuss this thing. Deuterion form. This is non-ionic form. You people know about all these things here. So peptide bond, you know, because next one or next today or next class I'm going to discuss about the what are the various hydration properties of protein and what are the covalent non-covalent interaction of the protein, so that I can easily move to the protein folding experiment and why uh, uh, what is folding and various other things, ubiquitination, that will be easy for me. So you people know about the peptide bond. I am not going to discuss about the peptide bond. It release two amino acid release water molecules that form is peptide bond. And you people know C alpha and C alpha carbon that I have already discussed. Most important that is, and these things also always remember, peptide bond absorbs the yeah, 214 nanometer in proteins, peptide bond, and but proteins we are seeing OD at 280 to 280. Protein Bradford we are taking OD as 540 or something because that is indirect method to detect the protein. If you have a pure protein, you have to take the OD at 280 nanometer. At what wavelength you are taking DNA? Anyone? 260
260
nanometer we
nanometers.
are taking DNA, and 280 we are taking protein, and 214 for peptide. You can ask me question, okay, sir, we have done Bradford 540, but it is indirect method of a protein estimation. So that we are taking, it depends on the how the that reagent is interacting with the protein. We are measuring indirect method, that is indirect. For direct method, you have to take the OD at 280 nanometer. So how protein is complicated? Just you think 100 amino acid long peptide can have 20 to 100 conformation. They can form the sequence 10 to the power 130 possible sequence means 1.2 into 10 to the power 13 Dalton molecular weight, and mass of universe is 10 to the power 80 Daltons. Okay, just you think how protein is delicate, how protein is complicated. Okay, universal like sufficient material to construct one molecules of each possible protein of 100 amino acid length. Just you try to understand these things, so that, because why I am discussing this thing, because next or after 2-3 class I am going to discuss about the protein, uh, uh, protein folding and all the things. You have studied little bit about the protein folding your second year or for third year? Anyone?
Yes, sir.
A little bit in proteomics.
Yeah. Have you heard about Annie-Fenson experiment?
No,
Sir,
sir.
could you repeat the name, sir?
Annie-Fenson experiment during the
No, sir.
folding.
No, sir.
Okay. Okay, fine. That is the basic. If you don't know Annie-Fenson experiment, it means you don't know the protein folding. So that things I am going to discuss. So just you understand that 100 amino acid long polypeptide, how complicated that protein sequence, and how there is a combination, permutation and mutation, permutation, and that how what type of conformation protein can form. Just see that 100 amino acid long polypeptide can have 20 to 100 10 to the power 130 possible sequences, and that molecular weight is equal to 1.2 10 to the power 134 Dalton. Universe lacks sufficient material to construct one molecule of each possible protein of 100 amino acid, so that we are saying that protein folding is it is a fraction of second process. And I will, in by any function experiment, I will prove, we will prove it is a guided process. It is a guided that. So okay, that will come later. These things I am not going to discuss. Protein is hydrophobic in nature, charge, polar amino acid. It can count 4 classes if glycine is considered its own class. 20 different amino acids are commonly abbreviated with 1 or 2, 3 codes. So polar, non-polar molecules, these things are important. Why these things I am going to discuss? Because when I will discuss about the protein folding and all, these things are very, very important. Polar molecules are generally able to dissolve in water. I know many students know what is polar, what is non-polar, but just to clear the basic, due to the polar nature of water, polar molecules are slightly positive and slightly negative charge. Example, if you table salt, ammonia, glucose, if you just uh, if you try to dissolve in water, it will dissolve easily. So these molecules are called as polar molecules, and most non-polar molecules are water insoluble, hydrophobic at room temperature. Example of household non-polar compound include fat, oil, and petrol. If you just put few drop of mustard oil or refined oil in water, it will float. That is the reason that that oil having a not a good friendship with water. Just you try to understand, if some molecules is polar in nature, it means it is hydrophilic in nature, it will like water and they have a good friendship with water. And non-polar molecules is a hydrophobic in nature. And so that last class I have discussed about little bit about serine and valine or alanine. Alanine is a hydrophobic in nature, so that alanine try to uh, and try to inside of the protein, but in case of serine, it is a what hydrophilic in nature, so it will always go to the surface of the protein so that it will interact with the water molecules. So these are these things also I'm not much I'm interested, because I know you people know about these things just for my course, so that you understand what are hydrophilic and hydrophobic molecules, because it will going to be come in during the next unit or after third unit also. So non-polar amino acid is a hydrophobic group: glycine, alanine, valine, leucine, isoleucine, methionine, proline, phenylalanine, tryptophan. One thing clear, I am not going to ask in your exam, you draw the tryptophan structure, you draw the phenylalanine structure. I'm not going to ask this type of question. Okay. So no need to draw this structure, but you have to remember what is proline, what is proline, what is tyrosine, what is tryptophan for your knowledge. So that is important. Then hydrophilic amino acid, it is serine, it is threonine. If you notice one trick, CS2OH. So we are saying that serine is hydrophilic in nature. Here also you can see OH molecules. It is hydrophilic in nature. Tyrosine also little bit hydrophilic in nature. OH group is there. Cysteine, that is make a disulfide bond. Remember pKr value is 8.4, pKr 10.5, and this is basic side amino acid, that is lysine, arginine, histidine. You people know about these things: aspartic acid, glutamic acid. So just you remember, if it is acidic, that pKr value is 3.90 and pKr for glutamic is 4.0. If it is a basic in nature, that pKr will be high, always remember. And histidine only, that is neutral, is 6.04 due to imidazole ring. But if somebody will ask you what is the pKr value of lysine, always remember lysine is basic in nature. If you want to guess also, it should be more than 7. Always remember, if it is arginine, you can say it is a more than 7. If you don't know the exact year, at least people will understand you have a basic knowledge about the amino acid. These are the ionizable group, lysine, those ionizable zinc 3 and all these things. I'm not going to. There is a one small exercise that is also important for your course. Whatever right now 2-3 class, all I am summarizing this course, okay, so that you people understand about the protein engineering. So a protein contain 100 amino acid. Calculate precise molecular weight of the protein if all amino acid present in the proteins are tryptophan molecular weight. Have you done this type of calculation in your first year, second year, third year? Yes or no? Student, yes or no?
Yes, a little bit in proteomics.
Hello?
Yes, a little bit in proteomics, sir.
Little bit on proteomics. Who taught you proteomics?
Sir, Kamalnathan, sir.
Kamalnathan sir. Okay, fine. So I'll just give you one small calculation. A protein contain 100 amino acid. Calculate the precise molecular weight of the protein if all amino acid present in the proteins are tryptophan. Molecular weight of tryptophan is 204. Can anybody want to try this, or I will just explain it? Yes or no? Shall I explain or
20.4 kilodalton.
hello?
Sir, 20.4 kilodalton.
20.4 kilodalton. Okay. Anyone other? No. Shall I explain? Yes or no?
Yes, sir. Yes,
Yes.
sir.
Yes.
Yes.
Yes. Okay. So a protein contain 100 amino acid. So as you people know the peptide bond. Okay. So peptide bond is formed between NS2 and COH. So when first amino acid, suppose first amino acid NS2 is here, and first amino acid, and when it will reach to 100, then COH will be free. So it means that one there is a one amino acid, one peptide bond will be less. You understand this my explanation? First you try to understand there is 100 amino acid, there is N terminal and C terminal. You know the protein having N terminal or C terminal?
Yes, sir.
Yes. So if there is NS2, if it is a circular peptide, then NS2, this NS2 1 and 100 COH will be make a peptide bond. But it is a not circular amino acid, a circular protein. So long chain of 100 amino acid. So that 100, 100, 100, 100 minus 1, then it will be 99 peptide bond. Correct? Correct
Yes.
or not? Yes. So when any two amino acid make a peptide bond, there is a loss of one water molecules. Correct or not?
Yes, sir.
Yes,
Yes.
sir. Yes.
One, yeah, 18, one water molecules will be loss of one water molecules. Then what will happen? 99 multiply by 18. So there is a 99 molecules, water molecules will be the lost. So that I have here, just prepare here. Total peptide bond, it will be form 100 minus 1, it will be 99. Okay, you people agree with this,
Yes,
student?
sir.
Yes,
Yes, sir. Yes.
because N terminal there is NS2, there is a CS2 that is flanking, that will not make an interaction. So that H2O2, as I said, peptide release. So 99 multiply 18. If you multiply 99 into 18, it will be 1782 Dalton. Clear? As that student said, it will be 20.40 Dalton. C is correct in this way, but C is little bit wrong, but her effort is very good. She at least she tried to solve the question. The molecular weight of 100 tryptophan is equal to 204 multiplied by 100 is equal to 20400 Dalton. Is it correct? Right now, you are understanding all the three part?
Yes, sir.
Yes. Then
Yes, sir.
molecular weight of peptide will be 20400 minus 1782. This is the loss, one peptide loss, because NH2SeOH is flanking, and 99 peptide bond multiplied by 18, 1782 Dalton is the loss of loss each peptide, 99 peptide. So that molecular weight is equal to 20400 minus 1782. So overall molecular weight is equal to 18618 Dalton. Are you understanding or not?
Yes, sir. Yes, sir.
Yes. Yeah, very good. But generally students are doing mistake here. Sometime in your GATE exam or CSIR exam, if it is a tip, if it is they have not mentioned tryptophan, or they have mentioned any amino acid, just protein or any amino acid, that time what you are going to do? If the question is like a protein contain 100 amino acid, calculate the precise molecular weight of protein if all amino acid present in the protein. Okay, just here, leave it here, delete this question, these things.
So, since we don't know what amino acids are there in the protein, how can we calculate the size?
So
So, if
we'll
we,
take the average weight of amino acids.
Yes, average molecular weight of the amino acid. Okay, understanding? What is the average molecular weight of amino acid?
110 Daltons.
Yes, that's correct. If there is a not, they have not given anything, then you have to calculate with 110 Dalton amino acid. Okay, here you people understand this question answer? Anyone having a problem? A protein contain 100 amino acid. Calculate the precise molecular weight of protein if all amino acid are present in the protein or tryptophan. Any question?
So,
Yes.
so in the question you asked just right now, if it's not mentioned what amino acids are given, so then how can we be sure that it is a precise molecular weight?
No, that is it is the rule that average molecular weight. Okay. It is question. It is not a It is just a question. Okay. If they have not mentioned amino acid, then you by default you have to consider as 110 is the average molecular weight of the amino acid.
Okay, sir.
It is a universal rule. Okay. In biochemistry, it is a universal rule. Okay.
Yes, sir.
Yes. Okay. Very good. So one more things that I am going to discuss here during the course also. Proline. Proline is formally not an amino acid but amino acid. So why proteins? When proline is in peptide bond, it does not have a hydrogen on the alpha carbon. If you notice that structure of proline, there is a in alpha chain there is a no. It will not form. Always remember proline. When proline is in peptide bond formation, it does not have hydrogen on the alpha chain. Here you can, it is alpha chain, hydrogen on alpha amino group, so it cannot donate hydrogen bond to stabilize a alpha helix or beta sheet. Always remember these things. It is often said inaccurately that proline cannot exist in an alpha helix. When proline is found in an alpha helix will have a single band due to the lack of hydrogen bond. Okay, so it cannot form the hydrogen bond. C alpha cannot form the hydrogen bond. So that many times if you heard it is called as helix breaker. Have you heard proline is a helix breaker?
Yes,
Yes.
sir.
Yes. So reason is that these things also I am going to discuss in protein folding chapter. So these things just for your clear, just for a concept that it does not have a hydrogen on the alpha amino group, so it cannot donate a hydrogen bond to stabilizing a alpha helix or beta seed. If you heard about the 3.6 turn and all the hydrogen, you have heard about these things or not? 310 helix and alpha helix beta sheet, you have heard about these things, no?
Yes, sir.
Because I'm
Yes.
not going because these things I'm not going to discuss in because this course is just for protein engineering. Just you remember in protein folding I will discuss about the proline thing little bit, how the it break the alpha helix and how the beta sheet and all the things. So if you see the structure of protein alpha helix, it will go the helix and suddenly it will convert into loop. If you notice that area, that place, you will see there is a proline or glycine. Okay, proline and glycine most probably present. So that proline is called as helix breaker. So these are the aromatic amino acid are relatively non-polar to different degree, and all aromatic amino acid absorb ultraviolet light. Many time if you remember what you are taking the OD of protein at 280. It is due to because at 280 tyrosine and tryptophan is and phenylalanine giving the fluorescence uh, responsible for most of the absorbance of ultraviolet light. So if somebody will ask you ki why we are taking OD at 280 nanometer, reason is that due to the tyrosine, tryptophan absorb more than do phenylalanine. Tryptophan is responsible for most of the absorbance of ultraviolet light by protein. Tyrosine is the only one of aromatic amino acid with a ionizable side chain, and tyrosine is one of the three hydroxyl containing amino acid. Due to this three three element, we are able to see the protein OD at 280. You can ask, sir, if there are any protein that is not contain tyrosine, tryptophan, or phenyl. There are some protein that having lacking of tyrosine, lacking of tryptophan, but most of the protein having a tyrosine, tryptophan, and phenylalanine. Any protein, most of the protein, 90% protein having a these three amino acid sequence, three amino acid. I'm not going this also much about this amino acid are in hydrolyzed form. I'm just going to discuss about what is the behavior of at PS7 amino group protonated in its anion form, carboxylic group unprotonated in its carboxylic ion form. These things. This is for also not for your this course, but I hope you people know about this thing. Just I am when at net charge, yes, one basic things. This always remember because these things also will come in during next class or after that class also. This never forget these three things. Structure varies with the pH. At low pH, all ionizable groups are protonated, and that will be cationic form. Always remember in case of amino acid. At mid pH, range of deuterion form dominant, and at high pH, the ionizable groups are unprotonated, or it will be in anionic form. Okay, so always remember when it will be pH will be 1. If the pH will be 1, what will happen? If pH will be 1, then what will be happen in the solution? Suppose if you are dissolving any amino acid which contain NH3, this one, NH2 and COH, what will happen when you dissolve any amino acid at pH 1? What is the meaning of pH 1?
Acidic pH.
Acidic pH. Acidic pH means
Higher concentration of hydrogen.
yes.
Higher concentration of hydrogen ions.
Yes, higher concentration of hydrogen ion. It means that that hydrogen will go to the when amino if it is a NH2 group, this one NH2 group, when higher concentration of hydrogen, then it will become a S3N. May I correct or wrong? Correct?
So can you repeat?
When if pH is 1, it means is acidic in nature. Okay, acidic in nature, and when it will be acidic in nature, the hydrogen ion is more. So to balance that, to maintain equilibrium, that hydrogen will be attached with the NH2 and it become NH3, and that this is called as cationic form. You are getting understanding or not?
Yes, sir.
Yes.
Yes, sir.
Okay, very good. So as you increase the pH from suppose that COH pK value is, I said 2.2, and if you increase 2.2, and what is the pK value of NH2? I told you, always remember these two things.
9.4.
9.4, yes, very good, correct. So when you increase the pH, suppose COH having a 2.2, when you are increasing pH 7, what will happen? pH 7 is higher than 2.2, then it is going to be protonated. Okay, always remember it will going to be protonated. When it will be going to be protonated, that it will be negative, and already because it is a this pK value of NH2 NS2 is 9 point something, so it is not going to be protonated, so that it will be positive and it will be negative. What is the net charge here? Can anybody tell what is the net charge here?
0.
Zero.
sir.
Very good. Plus minus 0. Now you are increasing, suppose it is a glycine molecule, now you are increasing the pH up to 13. Okay, pH 13. So when you will increase the pH, then what will happen? Again, there is a protonation or deprotonation. NS2, NS3 become a NS2. Okay, NS2 become a, it will be neutral 0, but here it will be minus. Then what is the net charge of at pH 13?
Negative.
Minus. So that at pH 7, this glycine will having a deuterion formation. I hope you have learned these things in your first year or second year of your biochemistry. I'm discussing these things. Same thing. Always remember this thing. At low pH, all ionizable groups are protonated. It will be in cationic form at low pH. Okay. Always remember that things. And at mid pH, it will be deuterionic form dominant, and at high pH the ionizable groups are unprotonated, okay, or deprotonated, and that is called as anionic form. It is negative in nature, like in case of it is same in glycine. If you want to calculate the pi value of glycine, then you can just here left and right 9.69 plus 2.3 divided by 2, it will be the uh, your pi value for glycine. Same things in case of uh, always remember only the ionizable side chain play a role in amino acid titration value. Okay, so I think you people maybe in your biochemistry in second year or third year, that faculty has taught you about this, how to calculate pi value. You know how to calculate the pi value, all student?
Yes, sir.
Yes, very good. So that is very good. So this way we can deuterion structure neutral value. We can, this will be in cationic form, it is deuterion form, anionic form. And like same way, like in case of leucine also, we can calculate this value. Then same way we have glutamic acid for 3 acid also we can calculate here, because same way here NS3 is not going to be, if you changing the pH 2.3, it is become a negative, it will positive, negative, higher pH it will be changed, and it will be -2, -1, it will be deuterion, here pK and here pK, you can join and calculate the, yeah, you calculate the pi value. These are just I have prepared for you people, building block of protein, no need of this. Just these are, you please remember, not need too much remember. Sometime I just need hydropathy index for your some, yeah, some course I need in between sometime, not always. Disulfide bond, just you remember, 16 residues form the disulfide bond. Two adjacent 16 residue can oxidize to form a disulfide bond. Disulfide bonds are usually found in the extracellular, not intracellular protein. Inside of cell is a reducing environment. Always remember, two adjacent 16 residues can be oxidized to form a disulfide bond. You people know these things?
Can you please repeat?
The disulfide, two adjacent cysteine residues can be oxidized to form a disulfide bond.
Yes. Sir.
Yes. Okay. Very good. Very good. So one question. if you know the disulfide bond, why outside protein, extracellular proteins contain disulfide bond? What is the reason? Intracellular proteins having less disulfide bond, no disulfide bond. What is the reason? You know the cell biology little bit? You people have a subject on cell biology?
Yes, sir.
Okay, just go through the cell biology course. Why extracellular proteins, they don't have a disulfide bond, but intracellular proteins, extracellular protein having a disulfide bond, intracellular proteins are not contain disulfide bond. Now always remember. What?
The cytosolic environment is reducing.
Yes, very good. Because now you are MSc student, nobody is going to ask you which amino acid form disulfide bond. Okay, any student can tell that cysteine is going to make a disulfide bond. Here you just see my slide, read my slide, whatever slides I am preparing. Disulfide bonds are usually found in extracellular, not in intracellular protein inside of cell. Because if you see in oxidation condition, that disulfide bond form. In reducing condition, disulfide bond cannot form. So that boy is correct. The inside of cell is a reducing environment, so that it not form the disulfide bond. Disulfide bond can be stabilized protein structure by providing the cross-linking, that is I'm going to discuss after next week or after that. Okay. So, hello. So you are getting the point?
Yes, sir.
Yes, always remember disulfide bonds are usually found in extracellular, not intracellular protein inside of cell is reducing environment. Okay. So significant of side chain also very, very important. Side chain determine the uh, solubility and all the things. And these are acid based properties of amino acid. I am not going to discuss, just I am uh, made a slide, you can go through it. If any student having any issue with this titration curve, then you can ask question, then I will explain. But for okay, that is yes. Protein have complex many ionizable side chain. I said that I'm giving one question here. Are you able to see the slide?
Yes, sir.
Yes. Can anybody tell me the timing? What is the time? Because I'm the slide full.
8:43, sir.
Oh, okay. I have to close here. Please, sometime I know I in classroom teaching. Sometime I take 1 hour also. So that please, because your school office said for not more than 40 minutes. So I am stopping here. Protein just you people. I am giving a one example. Uh, Here, hexapeptide, glycine, glue, aspartic, histine, lysine, alanine. Please try to determine PI of this amino acid. Okay. You people said you know that how to calculate the PI. So if you're not able to do, then next class I will. Okay, I will take the attendance. I will stop here recording.